Monsieur le Président, Mesdames et Messieurs, Madame Buzer, c'est un très grand plaisir de vous rencontrer. Je m'excuse que je vais parler en anglais. Je l'ai préparé en anglais la semaine dernière. Je n'ai pas pensé que c'était tellement français. Et bon, alors il faut vivre avec ça. Et boum, je vous demande votre pardon. The paths with Pierre Buzer and later with Arlette Rougeol-Buzer crossed twice in my life. And in both encounters were during a very decisive phase of my life. And I shall briefly dwell on the background because these two encounters and the embedding reflect interesting phases in the history of European neuroscience. At least I think they do. And they shed light on the development of Pierre's and later Arlette's scientific interests. Now, the catalyzer of our first contact was Otto Creutzfeldt. He was a close friend of Pierre, and I assume that the connection between the two was again catalyzed by Richard Jung in Freiburg, because Pierre Buzer was in the Black Forest in the laboratory of Cecil and Oskar Vogt for a while, which is very close to Freiburg, and at that time Creutzfeldt was at Freiburg. They must have met. And from then on, they remained colleagues and friends. My contact with Creutzfeldt occurred when I was still a very young student of medicine in the preclinical terms, because Creutzfeldt had organized, and I think this is quite sensational for the time, a seminar with an orthodox Freudian psychoanalyst, Professor Matusek, on the neurobiological basis of consciousness. And I thought this was fantastic. If one was able to bridge the gap between mental phenomena and the substrate of the brain by research. So I approached, and this was a wink of fate, not the psychoanalyst, but the neurobiologist, whether I could pass a thesis with him. And he said, yes, but you are still very young, you have to learn a lot, and maybe you come back to me later. And this was right at the dawn before I left for Cambridge to study medicine for 1 year. And I had already enrolled in Cambridge in a preparatory course. And then there were two losses that also determined bifurcations in my life. So there's a lot of serendipity in people's biographies. The first loss was that all my dossier that was sent from Munich to Cambridge got lost. And so I could not start my studies and would have to wait for half a year. So I decided I would not waste that time and try to study in Paris, because I was familiar with Paris, because I had done all my medical terms that I had to do during my preclinical studies in Hôpital Foch, in the service of Professor Guillot, who was a neurosurgeon pioneering stereotactic surgery. So I had decided to go to Paris. And then there was a second loss. On the last day of her stay in Cambridge, because her term was over, I drove a French student to London because she was to catch her plane and leave the island. And then a thief broke into my car, took her handbag, and there was passport, money, flight ticket, everything. And it turned out that she could not leave the island for 3 weeks. And after those 3 weeks, we got to know each other, and 3 years later we married. So losses are sometimes very important. So I inscribed as étudiant libre at the Sorbonne, it was still called at that time, and contacted Creutzfeldt again, asking him what should I learn and how and when and with whom. And so he must have contacted behind my back Pierre Buzer. And while I was doing my morning courses aux Instituts de Rue des Écoles, I got a letter. At that time there was no email, and students had no phones, of course. And I got a letter from Pierre Buzer inviting me to see him in Jussieu. He had at that time moved already from the Institut Marais, and he had invited me to participate in the Troisième Cycle de Neurobiologie, which he had inaugurated. I think it was a pioneering initiative to have a neuroscience curriculum at that time in France. I think it was unique. So he had already moved from the Institut Marais, leaving the Fessards, and founded, or he was the founding director, I think, of the Institute of Neuroscience at Jussieu. And at that time, there was an amazing density already of neuroscientists at Jussieu, which later wrote history. So I remember Michel Imbert, with whom Pierre wrote numerous books. There was Philippe Acher, who had not discovered the magnesium block yet. There was Alette Rougeol, who later should become your mother. I shall come back to her seminal contributions a little bit later, because I think one cannot honor Pierre without honoring Alette at the same time. There was Mario Wiesendanger, with whom Pierre had studied interactions between the visual and the motor cortex, so already at that time he was thinking in terms of systems. There was Michel Fabre-Thorpe, with whom he worked on the role of the thalamus in learning, showing that the ventrolateral nucleus was important for the acquisition of memories, but not necessary for the expression of the learned content. And then he continued, he was also collaborating later with Chantal Milleret, who is somewhere here, on interhemispheric communication, so again, a very timely systems approach. And still continuing to collaborate with the Fezards, and Henri had already mentioned this, they performed intracellular recordings, which was absolutely pioneering at that time. It was at the same time as Phillips in Oxford and Eccles in Canberra did this great seminal work. And actually Pierre had discovered what later became the complex spikes in the cerebellum. They saw these slow potentials but couldn't interpret them, but they had discovered them. So I think it was a great period of French neuroscience, especially in electrophysiology, molecular biology. Jean-Pierre had not yet conquered the field as much as it has now. So neurophysiology in France is intimately linked to names, and one has already been mentioned, Jacques Paillard in Marseille, Michel Jouvet in Lyon with his pupil Mike General, then Dora and Hirsch-Gerschenfeld, Ladislav Tauk, Robert Naquet and Yves Laporte at that time in Toulouse, I think. Then Tayarac, who had written the seminal atlas with Tourneau, and Pierre Buser collaborated with Tayarac, as far as I can recall, about once a week, obtaining recordings, intracranial recordings from human epileptic patients who were prepared for surgery. Also, seminal, very early on work that he did. And so when I let this, quand j'ai laissé ça passer revue, je me suis dit, mais c'est curieux qu'en France, il y a beaucoup de couples dont tous les deux sont des grands scientifiques, dans la bonne tradition de Pierre et Marie Curie. Et il doit y avoir quelque chose dans la culture française qui favorise ça. On n'a pas ça chez nous, for sure. So Pierre, and it was also briefly mentioned, he was in close contact with the international scene, which at that time still was a small and friendly and warm community, very much different from the competitive market that we are experiencing in the present time. It was the dawn of neuroscience, and it is associated, and I will give you some names just to evoke nostalgic feelings in those who remember those times still very well. Already mentioned Horace Magoon, with whom Pierre had worked in California and done important studies on the activation of the reticular arousal system through corticofugal input. I think it was his thesis together with Jose Segundo. Other people with whom Pierre had interacted, and one can find this in his biography, was Don Lindsley, Ted Bullock, Herbert Jaspers, Carl Leschly, Walter Penfield, Richard Jung, Walle Nauter, Mary Brazier, Hans Lucas Teuber, David Hubel, Thorsten Wiesel, Horace Barlow, Steve Kuppler, and Vernon Mountcastle. So those were giants, and they all had contact among each other, and they had influenced a lot also the work that Pierre had been accomplishing here. And so here I was, a very young, inexperienced young medical student, suddenly embedded in this very competent environment at Jussieu. And as I already said, after my morning activities at the Rue des Écoles dans les instituts d'anatomie et de pathologie, et mes stages à la Salpêtrière avec Arsin, à Sainte-Anne en psychiatrie, à l'hôpital Cochin en médecine interne, j'ai passé tous les après-midi dans les labos à Jussieu pour faire ces travaux pratiques et pour apprendre. Et c'est Pierre qui qui donnait des cours. Il était là. Il guidait nos mains. And it was absolutely amazing what we were allowed to do. He introduced us to produce spinal cord preparations in acute cats, which is a really difficult surgery. And he allowed us to do this, to get hands-on, to learn it. We performed intracellular recordings from spinal cord neurons. We had to build our amplifiers ourselves. At this time, no transistors, no computers. We had to have cathode follower electronics, so very basic stuff. So for me, this year was absolutely decisive for the rest of my scientific career, because when I returned to Frankfurt, to Munich, I got not only the courses that I took as étudiant libre recognized by the German university system, so I hadn't lost any time, but in addition, I had learned all the know-how I needed in order to start my thesis with Creutzfeldt on split brain preparations and EEG coherence. And this is why I'm really exceedingly grateful to Pierre Buser, who paved the way my way into neuroscience by accepting me in this course pro bono, which was absolutely against all regulations, I think. So he must have violated all rules of academic bureaucracy in accepting me. And I think this is worth a remark, considering the times it happened. This was 20 years after the end of the Second World War. And Pierre, I know, had suffered from German invasion multiple times, first when he was in Strasbourg, and then once and over again, his life was interfered with by our occupation. But he accepted me without hesitation and under extremely generous conditions. And the same was true for my fellow students. They were extremely kind and helped me to overcome my solitude, because at that time also my language was somewhat doubtful. But there was one problem, and I think it's worth communicating as well. Even though they're very close to me, they felt not able to take me home, because they felt that my German accent would evoke souvenirs of dark times, and they didn't want to have this imposed on their parents and their older generation. Because at that time, la traité de l'Élysée, ça faisait que 2 ans. C'était en 63. So now I would like to talk about the second crossing, which I think is equally important. About 20 years later, I had already moved from Munich to Frankfurt when I stumbled over the serendipitous discovery of these induced gamma oscillations. I think most of what I discovered in my life was by accident. But this made shift my focus of research, so I had to read other stuff than before. I was doing developmental neuroscience, which brought me close contact with Michel Lambert and Pierre Busseret here in Paris. And so I started to read about neuronal dynamics, systems physiology, and I stumbled over papers with authors Alette Rougeol and Pierre Buser. So I got into it, and then I saw, they had already were deeply involved in work with awake behaving cats, had multiple electrodes implanted over the cerebral cortex, and did a very detailed analysis of EEG oscillations under different behavioral conditions. And the way they did it was they partially showed a mouse to a restrained cat in order to evoke its attention. Genius. So the mouse kept on the tail, was squiggling, and the cat looked at that mouse trying to get it, and produced beta oscillations over the parietal cortex. Now we know that this is a neuronal signature of attention. They had also trained cats to do go, no-go tasks, so where they would have to withhold a response or perform one. And they saw that over the motor cortex, they had what they called the mu rhythm, a lower frequency rhythm, that always appeared when the cat was told to not respond, to withhold a movement. And this slow rhythm desynchronized when the cat was allowed to go ahead. Both findings survived the test of time. Very robust evidence has been brought up later. And so all of a sudden, these 20 years later, we got invited to the same conferences because we were working on oscillations and dynamics. And so we were colleagues all of a sudden. and discussed whether their beta was similar to our gamma or whether there were differences. So we were in a scientific discourse, but we also, what united us was that we apparently had been very far away from mainstream at that time, because the gold standard was single-cell recording of and analyze unit responses in the context of certain behavior, and the gold standard was intracellular recordings. So field potentials in EEG and all this was considered less serious. That has changed over the time. So it was a very long way from the first steps that Pierre had allowed me to go in his lab to the point where I finally was accepted as a partner and a colleague in the field of neuroscience. And these paths had miraculously converged 20 years after our first encounter, and therefore I'm extremely grateful to Pierre and Arlette. And I would be so happy if I could tell them directly, because we had then again lost contact, being busy everywhere. So, do I have another 2 minutes? Just to rush briefly through something that has changed since then. Let me see whether I can do this. Yeah. And you will immediately understand what progress is. The questions are still the same. The answers to them are still not available, but the methods have, of course, dramatically changed. So instead of putting a mouse in front of a cat to study attention, nowadays it's done in a different way. Look at that. You can't hear it, but the mouse is squeezing. So had they had this equipment, they would have done it that way for sure. Okay, let's stop this. Also, at that time, putting electrodes in the brain was a blind flight. We didn't exactly know where we were. Nowadays, one can use these non-invasive imaging technologies and know exactly, following structural and functional cues, this is functional MRI in monkeys and men, which functions are located where in the cortex. One can put electrodes there and study networks beautifully. Also, an enormous technical advance that was not available at that time. One can now implant microtechnology on the skull of animals and record for months, sometimes years, from defined populations of neurons, many 50 to 100, and get these very high-dimensional vectors that can be analyzed then with machine learning techniques, because now we have computers. At that time, there were no computers. If people wanted to compute the cross-correlation functions, they would have to print out the curves, mark the zero crossings, and then do a manual comparison and shift the two bin by bin by bin. So times have changed. So we can get these very high-dimensional vectors to analyze the language of the brain, which is distributed codes where most of the information is in the relations between the activity of all those neurons. We have a fairly good understanding of the connectome. We know which areas are connected to which. This is just a few of the connections in the monkey brain between cortical areas indicated as red dots. We can do the same with the human brain. We can look into it, trace the fibers with diffusion tensor imaging. And so we have much more possibilities to formulate educated hypotheses, because we can also set up wiring diagrams of the human brain and then record the activity of very much the same kind that Pierre and Arlette had already recorded, but we have more means to formulate testable hypotheses. We can train monkeys in a more sophisticated way than one can train cats, and record from them while they perform complex cognitive tasks. And as this slide shows, taking the signals that Pierre and Arlette had recorded 20 years ago, one can still get reasonable publications in journals like Journal Neuroscience, asking more or less the same questions: how does attention influence these wave patterns? So even though we have this enormous methodological progress, the progress in understanding is incremental. And I think, and he would be very pleased to hear this, analyzing these network dynamics is a very efficient tool for the analysis of disturbed dynamics in patients. And there's now a large amount of evidence that in schizophrenic patients and autistic patients, there are problems. This is just where one records the signals from it with magnetoencephalography. There are problems with the expression of these oscillatory activities that Pierre and Arlette had already described, changes in amplitude with simple cognitive tasks, changes in the precision with which they can synchronize over larger distances. And all those differences that can now be measured, they correlate with the clinical symptoms. And I think this is at least some progress, even though we have to admit that we don't understand at all what the basis of these diseases is. And I think I will leave it with this. Again, thanking very much you all for having me here, and to my, as you now know, real scientific mentor. He started it all. Thank you.
Thank you very much. And is there any comments, additional remarks, Henri?
Yes, thank you for this, these comments on the 40 hertz that Arlette, Rougel, and Buzer discovered when they call it beta.
Beta,
basically, yeah. It's known now as the gamma oscillations.
No, there's a difference.
Basically the same.
No.
So
no,
in what way?
I skipped a few slides, but they had rightly called their rhythm beta. It was between 15 and 30 hertz or so.
No.
Sometimes it was higher up.
It was higher.
But they found it over areas that we are now...
frontoparietal cortex
parietal,
of
prefrontal.
cat and monkey.
Yeah. In monkey, the frequency is higher.
Yeah.
In cat, is a bit lower. And this is the beta oscillations that are the signature of an attention network that comprises frontoparietal cortex.
Right.
Now, the gamma is something different. It's in a higher frequency range. It's mainly induced, so you have to drive the neurons. It can also come top-down. You can do gamma feedback, and then people learn to produce it. But it's clearly distinct rhythms, whereby the gamma rhythm is more the conveyor belt for feed-forward information, while the beta rhythm is more the conveyor belt for the top-down information. So the two are interdigitating. But it's reasonable to keep them apart.
Well, possibly. Never mind. If so, could you add a little bit more about what is specific to the gamma oscillations in schizophrenic patients and autistic patients?
What we see is...
Yes, I'll be interested to know what's new.
Yeah. The power of the oscillations... is of the induced oscillations during a cognitive task is strongly reduced. And this reduction is proportional to the severity of the clinical symptoms. But the most striking finding is that the ability to synchronize the oscillations in phase is reduced, dramatically reduced.
You mean between different areas?
Between different areas,
okay.
yeah. So this is the leading symptom, and this is very similar in schizophrenia and autism. And strikingly enough, something very similar happens during the development of human children normally. When they, we did a longitudinal study, when they go from post-puberty to early adolescence, at this transition phase, they have already built up a network that is reasonably precise and has reasonably high frequencies. And this gets completely dissolved for about 2 years before it reconsolidates and creates a network that is more focal, more precise, and then adult-like. And at that developmental phase, children look like schizophrenic patients. If you would just look at the electrographic signatures, you would be worried and say, this is a clinical case. This is beyond puberty. At the time where, if schizophrenia breaks out, it tends to break out, 17, 18, 19. So there's another late critical period in human brain development that needs to be explored.
Okay. Hopefully.
Merci. Oui, pardon. Philippe?
Yeah, thanks. Coming back to the issues of beta and gamma oscillations, Something that we see very frequently in humans is that each time you see a gamma oscillation at place, you see a beta decrease, a decrease of the beta rhythm. Do you have any explanation for that?
Yeah, there's different pacemakers, and apparently they compete with each other. So it's a general phenomenon. If you have increase in gamma, you have a decrease in the more lower frequencies, except theta. They can coexist. Then gamma rides on top of the theta oscillations. But why this is a general principle, I don't know.
It's always very interesting to see how a mentor can influence someone. Could you tell us how Pierre and his wife did influence your own view about your very good research?
Well, you know, I was very, very young when I was there, and I was trained to do spinal cord surgery. I think there were two more things at the meta level that I learned. The first was generosity, and the second was to overcome our history. After all, I was German and he was French, and he had suffered. He came from Strasbourg, and I was integrated as if I was part of the family. So I think there were these two experiences that were very basic to me. And the third was the hands-on lab work. I learned that you could build your stuff yourself and not be dependent on ready-made consumables that one can buy and not understand what they really do. Pierre forced us to really understand what the resistor does and what the tube does. So it was grassroots physiology. And I think this primed me my lifelong. The scientific contents, I was too young. I didn't understand much of the brain.